Hi, I'm Aditya. I'm studying biophysics. In this video, I want to share what I do in my lab. But before that, we need to understand what biophysics is and what does a biophysicist studies. Our universe is full of marvels, from black holes to human brains. But for a biophysicist, the phenomenon of life remains in the spotlight. Biophysicists try to decode the physical laws that govern life. We study living cells and their individual components using our fancy instruments. But what exactly are these components of life? You might have seen them in your high school textbook, which something looks like this. But in reality, a living cell is much more intricate and crowded. To understand this better, let's go outside. So if we consider this whole place a living cell, then these people, including me, would be its components. And these big stable structures that you are seeing would be the cell's organelles. There are so many unique components that makes the process of life really complicated. To understand the intricate mechanism by which a living cell functions, it's really important to observe just a few molecules at a time. If a scientist tries to study just one molecule, like me, out of so many molecules, it will be really hard, as there will be noisy signals from so many molecules. But scientists have invented new techniques to observe one molecule at a time, and they call these single molecule techniques. These can be broadly classified into two categories, fluorescence-based and force-based. In fluorescence-based techniques, scientists put dyes on the molecules, and as they shine light on these molecules, they start glowing, allowing scientists to observe molecules one at a time. Whereas in force-based techniques, instead of just watching them, scientists can touch and manipulate the molecules. A molecule can be in different states, and on applying force, it will react differently, which can reveal some important information about its states and its behavior. So I use one of the force-based techniques using this instrument called atomic force microscope, in short AFM. But what do I study? Well, I study one of the most interesting molecules of life, proteins. But why proteins? You have probably heard about proteins from gym trainers or food labels. But proteins are much more than just dietary components. In the microscopic realm of life, you will find proteins playing a starring role. For example, take hemoglobin. It transports oxygen and carbon dioxide in our bodies. And like hemoglobin, there are more than 20,000 proteins inside a human body performing different tasks, just like a miniature machines. And if we consider all living species, then this number will reach into the millions. But how can living organisms can make so many different protein molecules? Well, there is a secret recipe to it. If we zoom in a bit in this protein structure, we will find out it is made up of amino acids, and they come in just 20 different flavors. And these 20 amino acids can be linked together in different sequences to create millions of different proteins. Just like with only 26 English letters, you can create a million-word story like Harry Potter. But here is where it gets interesting and a bit tricky too. Knowing how a protein will look like after its amino acids get linked, it's a million-dollar problem, also known as protein folding problem. Scientists from many different fields are trying to solve this problem. Recently, using artificial intelligence tools like Rosetta 2 or AlphaFold 2, scientists have started getting some structural information of proteins. But picture is still incomplete because proteins are not just static, they are vibrant. They are constantly wiggling around, going from one state to another. That's where tools like AFM comes into play. With AFM tip, I can delicately interact with a single protein molecule, just like a nanoscale puppeteer. I can pull on a single protein molecule to watch it unfold, and then releasing the force to watch it refold. As we apply force and release force, we can get a glimpse of different states of a single protein molecule. Through the AFM, I get to witness the fascinating dance of proteins. It's a dance of one of the most interesting molecules of life. And I am on a journey to uncover the mystery of proteins, one molecule at a time. Hey everyone, my name is Aditya and I'm getting trained to become a scientist.
So today I'm going to talk about Okay, so today I'm going to talk about what do I do in my lab, how do I do it, and why I do it. So answer to the first question is pretty simple. I take protein molecules and I treat them like a spring and I pull them apart. And I do it like one molecule at a time. So the bigger question is, how do I do it? Well, I use this fancy instrument called AFM. Why this is fancy? Because behind this eyepiece, there is a place where I can put this tiny little tip, which is so tiny, like 1,000 times thinner than your hair. And I can use this to pick up only few molecules at a time, and I can pull them. So that's how I do it. Well, now the biggest question, why I do it? Protein being a biological entity, why would someone wants to pull it? Well, I can give you my personal answer to it. So let's start from the very beginning. Well, I never liked biology because this diagram used to scare me. How am I going to remember all of these names and their structure and their function? So I never choose biology. I dropped out from the biology in my high school. But something funny happened. If I'm not doing AFM right now in my lab, 90% of time I'm doing biology. I'm either cutting, pasting DNA from one place to another place, or I'm making bacterial cultures. Well, oh, sorry, that's not me, that's me. I'm drawing, so somehow biology chose me. But how did it happen? Well, answer lies in my college days. So I chose chemistry during my undergraduation, and I found that, okay, these molecules, these biological molecules, are made up of these organic chemistry textbook. These comes from organic chemistry, and not only lipids, but nucleic acids, which are DNA and RNA, are also part of these chemistry molecules, simple molecules. So okay, I thought, okay, so maybe biology is not that hard if it is can be understood by these 4 classes of simple molecules. Okay, so I started biology again, but I was wrong again because this is an image of nucleus of human cell nucleus, which is million times magnified, and you can see there's so much crowd and many things are happening. These are so many molecules and life is complicated. Okay, but we can all appreciate the fact that these all molecules are coming together to keep us alive. And what fascinated me a lot, if you remove these darkened area, most of the part are the proteins. If you take a rough estimate, then the dry weight of a cell is 70% of proteins. Okay, and these proteins does all the work inside our body. If you look at this particular molecule, this does very cool job inside our body. It takes proteins from one place to another place. And like these, there are many other proteins which does all the work to keep us alive. And whenever I think of proteins, I think of personality because they comes in different shapes and sizes. And they have their friends, acquaintances, even sometimes enemies as well. But being organic chemistry molecules, how do they do it? They are not conscious. They are not conscious. So the answer lies in their surfaces. If you see, they have these groovy surfaces on them. So whenever, just like key and lock, they have groovy surfaces, they come together, and whenever they come together, they click, and something happens. Just like these protein molecules also have these groovy surfaces, and whenever they come together, they start grooving together, just like these two molecules are grooving together. But sometimes some bad things also happen. For example, there can be heat and heat stress inside our bodies. So what does cell do? It comes with a protein molecule which can save it. So, hello everyone, I am HSP, and whenever there is whenever there is heat problems, I come and rescue all your proteins from getting melted. But today I have a complaint to you. I do this job day and night, but I don't get anything to do it. Can't I have some dreams? Because since I was born out of my mother ribosome, my father came and decided, okay, you will become a firefighter. Mera beta firefighter banega. And my aunt tRNA named me heat shock protein, so that's why I got this name, HSP. Since then, nobody asked me what do I want to become. Okay, and today also nobody also wants to listen to me. Is there anyone who can listen to me? I am being a scientist. I will listen to you, HSP. Don't worry, I am here. So, what were you saying? Sorry? Sorry, I'm not able to hear you, HSP. Can you speak? Okay, sorry. It's little loud, noisy there because there are so many protein molecules all talking together so his noise is not getting audible to me. So we have to find this solution. Okay? Maybe I have a solution. If somehow we can isolate it and we can study in the isolation, then maybe we will be able to study, we will be able to listen to HSP. These techniques are called single molecule techniques. And if you remember from my house slide, AFM is also one of those single molecule techniques. So maybe I can use that to listen to the story of HSP. Maybe then he will be happy. So let me use my single molecule technique. Okay. Now HSP, you are alone and I will listen to your story. Don't worry. So it's not like that I hate my job being a firefighter. It's a noble cause because I can save my fellow proteins. But there is no adventure in it. I want some adventure in my life and I click really good pictures. If you see, I go in wild and I click these really cool pictures. So I want to become a photographer, but I don't know how can I become this photographer. Since I was born, I was given this only job that I can do. I have no idea. Can you give me some idea? Okay, being a scientist, I will help you. So the solution lies in your groovy surfaces. If you remember, if you can change these groovy surfaces a little bit, you can attain a different state. And one state can go to a firefighter, and if you go to a different state, you will become a wildlife photographer, and you will be able to do 2 jobs at a time. You have to just switch these states. That's all. That's all I do in my lab. I take different protein molecules. Just like HSP, there are many protein molecules which wants to share their story. So I go near to them, like very close to them, like literally touching them, and I try to study their different states. As HSP had different states, every protein molecules can have different states. So that's how I study them, and I try to study. So with AFM, I get to witness different states of a single protein molecules, and these are some of the really fascinating story of these proteins. And I am on a journey to study these protein molecules one at a time. Thank you.